All right, so let's talk about the cell structure functions and its and the different way of transporting across the membrane. In in another way, usually I'm going to talk about the plasma membrane and the way to transport across the membrane in my next chapter. So here I just want you to introduce you to the plasma membrane. When I talk about the cell structure, of course, it will include this plasma membrane. And this is the one that really control the transport, control what is coming in and out. We will talk briefly about it, but we will talk deeply on the next chapters. We will focus most of it on the cell structures and functions. So let's give some history. Galileo, he's the first one that really develops a microscope compound with a convex lens. Robert Hooke, this Belgian gentleman, he was looking at using this microscope, looking at a cork bark cell, and he looked at it and he said, "Oh, such a cilia, such a compartment." Therefore, the name of cells came up. Later on in 1600, using an handcraft microscope, he observed a single-celled organism that he called animalcules, and which on our day we call microorganisms, such as bacteria. And in the mid 1800, Schwann, the Swedish, I think he was Swedish, he observes the first one that observes animal tissue. And Virchow, 55 years later, he's the one that said that the cell, he studied the cell reproductions, and he came up with the idea that every cell came from pre-existing cells. So therefore, we are going to develop what we call a cell theory. All organisms are made of one or more cells. That's one. Cells is the basic unit of life. All the organism, all the living thing, we are united, we all are made of cell. If not, we don't call them living thing or organism. And all cells arise from pre-existing cells. Not many cells can be seen by our naked eyes. Okay, we can't. There are some bacteria, there are some viruses, like the viruses around us. We cannot see it with our naked eyes. You see how naked eyes can see it? You can see from 100 micrometers, like to up. That's, but anything that it's under 100 micrometers, we have to use a special microscopy to see them. But even though some microscopies are very limited, for example, the one that we have in our labs here at our campus, it's what we call light microscopy. Light microscopy can see things like a little virus, but even that we have to use like a special contrast to see it. And to see, for example, we cannot see atoms or lipids, we cannot see proteins, but with the light microscopy we see it. So the light microscopy, I believe you already studied the anatomy for it. That's the ocular, the objective, I mean the amplification for the ocular is 10x. That means it amplified 10 times whatever we are looking in the stage, using different objective that we have here in the nose piece. The scanner is 4x, the lower power is 10x, the higher power it's 40x, and we have the biggest objective is the one that's amplified 1000 times. It's the 100x, or oil immersions. So how that happen? How we can see actually and amplified any samples we want to study to see it, any cells, for example. We have to use, we are using just the light, right? So therefore, we are using a piece of glass that is transparent to the light, that we call slide in our lab. And a lot of times we have to cover it with a cover slip. Therefore the light coming from both the bulb for the microscope is going through, and whatever objective we are seeing, it will be amplified, and we have to multiply this by the 10x of the ocular. So the quality of the image depend on the magnification, but not only that, it depends also and the contrast. In the contrast is how the specimen that we are studying in our slide stands out against the background. And I give you an example. Like, for example, here, this is end-stain specimen. I can see the border, I can see it, but it's very hard with my eye, because I am very experimented a cell biologist person. I can tell this is the nucleus, but for a naive eye, if I told him where is the nucleus, that's the nucleus. Here we stained. This is what we call contrast, a specimen. So we stain it with the methylene blue, and the DNA that is in our nucleus take off this methylene blue and making it darker than any other part in the cell. So we use contrast, that means how the specimen stand out against the background. And we do also some magnification, you understand it. Contrast, you understand it. Resolution. Resolution is how we can clarify the image that we are looking in through our ocular, in our field of view, neat clarity. That means how we can have the capacity to separate a 2 point in this space. And these resolutions, we use different things. We use the iris to reduce the light. We use the fine knob. We use the coarse knob. Electronic microscopy, it's much bigger than light microscopy. And instead of light, we don't use light, we use electrons as beam. So there are two types of microscopy. We have microscopy that focus only on the surface of a specimen, providing kind of like 3D, three-dimensional image that's scanning electron microscopy. And we have a microscopy that the beam of electrons, they go through a specimen to study actually the ultrastructures, the internal structures of the cells. This is transmissions microscopy, and this is a picture of a light microscopy. And since we use electrons, we don't use actually a slide, a piece of glass. We use a little a circular teeny a slide of gold. All right, and then we have how many type of organism? We have prokaryotic organism, and we have eukaryotic organism. Therefore, we have prokaryotic cells versus eukaryotic cells. What is the biggest difference? Prokaryotic cells, the biggest difference between prokaryotic cells and eukaryotic cells is that prokaryotic cells, such as bacteria, coronavirus, bacteria, coronaviruses, flu viruses, they do not have a nucleus. They don't have nucleus. Their material genetics is not inside of a nucleus. They don't have any membrane bonded organelles. The eukaryotic cells, not only they have the DNA is inside of the nucleus, within a double membrane around the nucleus, but it's also almost each organelles have a membrane banded organelle. Like Golgi have a membrane around it. Reticulum endoplasmic have a membrane. Even lysosomes, those little vesicles, they have a membrane around it. So it's look like eukaryotic cells has like a little compartment a little compartment. This is why we call it compartmentizations. Their size is much bigger than prokaryotic cells. They reproduce sexually and also asexually, eukaryotic cells. Prokaryotic cells, they reproduce by binary fission asexually. Okay, and then this is an example of prokaryotic cells. You can see that we have, this is the three things that all the organisms they have. You know, the cells unified all the organisms. All the organisms are made of the cells. And not only that, all the cells for all the organisms, they are made of three things: material genetics, DNA, plasma membrane or cell membrane, and ribosomes. Three things that you are going to find commonly existent in each cell. All the cells, eukaryotic, prokaryotics, they are all made of cell membrane, ribosome, and material genetics. Okay, so this bacteria, their material genetics is not included in the nucleus, but they have a space where it's localized. It's called nucleoid. This bacteria, it's a plasmid, by the way. It's a DNA plasmid, DNA bacteria. So their bacteria is within a space that we call nucleoid. They have ribosome, those little dots over here, to produce proteins. So they need DNA from material genetic, and they need a food from ribosome, and they have a plasma membrane as a fence to protect the cells. But this is a bacteria, have also a cell wall, and in the top of the cell wall, this is a cell wall. They have a capsules, and this extension of the plasma membrane. They are also, this is dosapili. This is an extension of the plasma. If you look at the plasma membrane, is going out. You see, those are extensions of the plasma membrane. They are not extension of the capsules or the cell wall. It's a extension of the plasma membrane. Okay. And those capsules actually, because bacteria, when they enter cells, they are tendency to be attacked because the cells have a way to protect itself from this microbial agent that is bacteria by phagocytosis. So those capsules is to protect them from getting eaten by phagocytosis. Eukaryotic cells are much bigger, all right? They have a true nucleus. They have many organelles. So we are going to focus on the anatomy of animal and plant cells. That's an animal cells and that's a plant cells. Just the morphology, if you look at it, the animal cells as like a circular globular, and look at the geometric form of it. They can be like this, they can be trapezoid, they can be square, they can be rectangular, but animal cells in general, they are like circular globular. So let's look at each structure. So we have a plasma membrane that I told you that all the cells have it. So let's look at what the plasma membrane is. So this is the plasma membrane. Okay, that's yellow things that you'll see over here. That's a plasma membrane. It's the fence. It's the one that protects the cells. We do have ribosomes, those little dots that you are going to see. They are red. You can see them either free in the cytoplasm or either attached. Actually, look at them. They are also over here. They are over here. And material genetics. Well, this is eukaryotic. So the material genetics, which is here, it's inside what we call a nucleus. Okay. Chromatin is a DNA. It's a material genetics. Okay. It's a DNA plus a lot of protein on the top of it that we call it chromatin. Okay. So we have the nucleus. Inside of the nucleus, as I said, there are nucleolus. we have chromatins, and one thing's very nicely, not only we have one membrane around the nucleus, but we have double membrane. So we call it nuclear envelope. Nuclear envelope. Okay. So the job for the nucleus here, I can show it to you. That's the nucleolus. So the nucleus stores the chromatin, which is the DNA plus proteins. And it's within a space, this space inside, it's like a jelly-like substance. We call it nucleoplasm, the inside of the nucleus. And then we have the nucleolus. This is actually very condensed region of this chromatin that you are seeing over here. It's very condensed. And this is responsible to produce ribosomes, those little red spot or dots over here that you are seeing, like ladybugs, dots, red dots, they are produced by the nucleolus. So the boundary for the nucleus, as I said, is not only one membrane, but it's a double membrane. We call it nuclear envelope. Nuclear envelope, if you can see, we have one over here and another one over here. Nuclear envelope. Okay, I will talk to you later on about the plasma membrane, but I think we already know. And I remember we done even a little sketch about how the plasma membrane is made of. Is made of a, a bilayers, double layers of a phospholipids. Okay, we will show you what phospholipids are. One thing is also that we are going to see in the nucleus, not only he store the material genetic, right? But also he is very close to another organelle. This organelles, we call it reticulum endoplasmic. We have two types of reticulum endoplasmic. If you look at very close, we have those reticulum endoplasmic which are completely covered by this red dots that are ribosomes, and we have some reticulum endoplasmic that they don't have those red dots on them. So therefore those are called smooth reticulum endoplasmic or endoplasmic reticulum, and those are called rough endoplasmic reticulum. Smooth versus rough. The rough reticulum endoplasmic or endoplasmic reticulum, their job is to secrete protein. The smooth reticulum endoplasmic, they are responsible to synthesize lipids, entre parentheses, fat. This is why they are smooth. And they are also playing a role there in a cell detoxification and muscle contractions. If you realize that the liver are very, very rich on smooth reticulum endoplasmic, this is why we have a lot of fatty acid in the liver. And the first organ that shut down when a person is a drinker is the liver, because it plays a role on detoxification to take away any toxins such as alcohol from our body. So when somebody is drinking too much alcohol, the liver that is very rich on the smooth reticulum endoplasmic is going to shut off because he's playing a role on cell detoxification. The next organelle that we are going to study is the plasma membrane. The plasma membrane is a bilayer of phospholipid. Phospholipid is a lipid. All right, it's a I have a phosphate group, that's the head that you are seeing here in orange or red over here. And those two are two fatty acids. Two fatty acids, and they are they make what we call phospholipid bilayers. But not only, that's the major constituent, but you can see over here some blue proteins also. So we have those channels proteins, we have those peripheral proteins, we have also those integral protein. If you look at them, they cross the membrane. That's mean they are made of some amino acid that they are polar, that they can interact with the phospholipid over here. Because the phospholipid, this part is hydrophilic, he interacts with water, and this part over here in the middle does not interact with the water. This is hydrophobic, hydrophilic, hydrophobic. And it's so funny because the plasma membrane, the cells, they are made almost, I mean, of water, 90% of it. So we have outside is water, and inside of the cell is also water. So therefore, those phospholipid head, they are going to be in contact with the watery part. This is why you will see them in contact with the water, because inside is water, outside is water, but between, this is the fatty sandwich. So those proteins actually that cross the intercross this membrane, they are therefore made of two types of amino acid that they are polar, that they can interact with water, and amino acid that they are non-polar, that they can interact with the fat, because fat is hydrophobic. The water rejects hydrophobic substance. What else we have? We have cholesterol. Later on, when we study the next chapters, I will teach you what's the job of this cholesterol. It's to maintain the fluidity of the membrane. And not only that, look at that. We can find some sugar. Those are sugars, carbohydrates. When they are attached to those phospholipid, we call them glycolipid. And some of them are attached to proteins. You see that? Those are called glycoproteins. Glycoproteins. Glycoproteins is sugar plus protein. It's a complex between sugar and the protein. And this one is between sugar and lipid. This is lipids. Those redheads are phospholipids. So this is why we call it glycolipids. Okay. And the function of the plasma membrane is to control, to really screens on what is get inside and what is go out from the cell, in or out. Even a molecule of water cannot cross the membrane easy. Imagine I am a molecule of water and they want to go from outside to inside. I will have no problem to interact with the head because it's hydrophilic. It's a water loving. He has affinity for water, but I cannot cross this hydrophobic sandwich of fat over here. I will be rejected. Okay, so I cannot cross. All right. And next is genetic information. The ribosome. The ribosomes are the site to produce the proteins. They are made, I mean, by two subunits. We have a large subunit that you are seeing over here on red, and you have a small subunit. And in the small subunit, we have actually an active site to bind to a messenger mRNA. They are very, very important on producing proteins. I will not enter in details on it because we are going to study that in our last chapters during the gene expression. So the ribosome really play a major role during this gene expression to produce the proteins. Before I go to the next step, which is the endomembrane system, I want you to focus on this slide. If you look at this rough reticulum endoplasmic, it's in continuity with the nucleus. You see it? That's the envelope nuclear, and it's in continuity. Envelope nuclear, it's in continuity. And it's also in continuity with the smooth reticulum endoplasmics too. Not only the rough, it's also with the smooth. Okay, so those endoplasmic reticulum, both of them, they are part of what we call a system of intracellular organelles that deep operate together. We call it endomembrane system. They either communicate directly physically or they communicate actually by vesicles. They communicate by vesicles. Look at that, the Golgi with reticulum endoplasmic, they communicate through vesicles, and the Golgi communicate with the plasma membrane through vesicles too. So this is why I'm telling you that they are part of what we call endomembrane system. We have also an endomembrane system that is the Golgi Apparatus, and we have also some lysosome and the central vacuole. The central vacuole exists only for the animals, not for plants. Okay, so the Golgi, if you look at it, he have two faces. He have a face, and I am going to show it to you in a big picture. You have a face that really is directing, looking at the nucleus, and we have the other face, the opposite face, is looking away from the nucleus, is looking at the plasma membrane that way. This is the Golgi, okay? So the face that is looking at the nucleus, we call it a cis face. This is the one that will receive vesicles from the reticulum endoplasmic. And this one is actually the shipping phase, is the one that will secrete uh, secretory vesicles to go to the plasma membrane, for example, over here. So that's the trans phase, and that's the cis phase. The job for the reticulum endoplasmic is to receive and modify and store and ship product from the reticulum endoplasmic. So he play a role like a post office. He receive and ship product, but he also modified, like for example, if some proteins came in and some carbohydrates are in, they can make glycoproteins, and the proteins over here become more matures before they get shipped anywhere in the cells, that it's a part only of the endomembrane system. He's not going to ship, for example, to the mitochondria, because the mitochondria is not part of the endomembrane system. Lysosome and vesicles, they are part of the endomembrane system. They are coming out from the Golgi. Lysosome, they are hydrolytic, acidic, because they contain enzymes, digestive vesicles. They contain enzymes that break down macromolecules, and they also play a role not only to transport but also to protect the cell against any any microbial agent, and this is by phagocytosis. Phagocytosis means eating and breaking down. That means the vesicle will eat, for example, the bacteria will put it inside, and the enzyme that they are inside of this vesicle will break this bacteria down and will kill it, of course. So let's look at this. So we have, for example, we need, let's see, we need to transport outside some content. Okay, So we came in from the reticulum endoplasmic, go inside the vesicles. Information came from the DNA, all right? Go through the reticulum endoplasmic, go through the vesicles. The vesicles came to the cis face and fuse with the cis, the face of the Golgi apparatus. He go through a lot of process inside of the Golgi that we call it cisternae. And then again, another vesicle is going to transport this modified content. Example, let's see, it's a protein, for example, and you will pack it into vesicle, and the vesicle travel to the plasma membrane, releasing its content outside in the extra environment, cellular environment. We do have also the central vacuole. It's only for plants. This central vacuole accumulates water and helps the cell elongate. He stores also iron and some nutriments, some organic compound, and some soluble pigment. All right. We do have peroxisomes. Peroxisomes, the oxidized amino acid and fatty acid, generating toxic hydrogen peroxide, and those toxic hydrogen peroxide are going to be catalyzed by hydrogen peroxidase, which is a catalyze, which is an enzyme, and you are going to see some crystals inside of those peroxisome because they are very rich on this enzyme. Okay, so I go back to my cells. So we cover what is nucleus, we cover the smooth, we cover the reticulum endoplasmic, ribosome, plasmamembrane, peroxisomes. Now we are going to talk about the mitochondria, and we cover also the central vacuole for the plant. Okay, so it's the same for we cover each organelles that they are so far: the nucleus, the reticulum endoplasmic, the Golgi, the central vacuoles. Now let's talk about the mitochondria. The mitochondria exist in both animals as well as plants. The mitochondria is the site for cellular respirations. It's the one that it's going to harvest the energy from your organic fuel, from your food, to transform it on a chemical energy under the form of adenosine triphosphate ATP. Because your cells, to do the job, the only energy we recognize is under the form of ATP. In another word, the mitochondria is able to harvest energy from your organic fuel, which is your food, and store it into ATP. And ATP is the energy that the cell will recognize. It will not recognize the food you are eating. The only energy that the cell will recognize is under the form of ATP. All right, so what is the anatomy for mitochondria? So we have also double membrane. We have an outer membrane, which is the one that is in contact of the cytoplasm of the cell. And we have an inner membrane, and the inner membrane fuse inside, invaginate inside to form what we call cristae. And the inside of the mitochondria, we have a matrix. One thing is very, very funny, very, very nice, is that the mitochondria is made, has his own ribosomes. And not only that, he have also his own DNA. Mitochondria, it's not part of the endomembrane system. So it is a factory of ATP, and not only that, It also plays a role in apoptosis, which is the programmed cell death. The cell will die, but it's a program. That means there are a lot of, it doesn't die just like that, okay? They have like a signaling pathway for that also. Chloroplast. Chloroplast, this one, mitochondria, you can find it in both animal and plant. The chloroplast, you cannot find it in animal. It's only in a plant. And I go and they show you in the plant. We go back to our original cell. These are chloroplasts. Those strings that you are seeing greens over here, those are chloroplasts. Okay? Chloroplast is the site for photosynthesis. It's an organelle that is important because the plants are the producer of the planet. They are the one that will uh, capture the sunlight, right? And transform and produce an organic molecule, an organic fuel. They are the producer. Without the plant, we cannot survive. Those chloroplasts contain a pigment that we call chlorophyll. It's always green color. It's very important for photosynthesis. Chloroplast is also made of a double membrane. He have an inner membrane and outer membrane, or internal. They call it here internal or inner, and we have between an internal space. Okay. The mitochondria also has between the outer and the inner membrane and internal and intermembrane space. Inside, we have what we call the thylakoid. Those thylakoids are a membranous sac within the inner membrane. They are membranous sac. When we call it a thylakoid, more than one, we call it granum. One is thylakoid, more than one, we call it granum. Here also, you can see DNA. Chloroplast has DNA and has also those little red dot, that are ribosomes. Inside of the chloroplast, we call it stroma. Stroma. For the mitochondria, we call it matrix. Here we call it stroma. For the nucleus, we call it nucleoplasm. For the entire cells, we call it cytoplasm. The stroma is the internal fluid. And the chloroplast is just one of the group of plant organelles that we call actually plastid. Here we are, I love this slide because it summarizes the function for each organelle. I want you to go through it and copy it by your rating hand. All right. Another organelle that exists in both cells, either plant or animal cells, this is what we call cytoskeletons. The cytoskeletons are those organelles that will maintain the cell shape. We have the microfilament, the intermediate filament that appear over here, little purple, and we have the microtubules that appear over here, yellow. Those yellow microtubules. Okay, so those, exactly like the word said it, they are cytoskeleton. Cyto mean a cell, skeleton, it's the skeleton, is to maintain. This is a system of proteins, fibers that are going to maintain the shape of the cells, and therefore they are going to provide structural support, and in some cases even locomotions, um, I mean mobility for the cells. I love that pictures. Look at all those microfilaments. We have the intermediate filament, the microfilament. and the microtubules are those green ones over here. This is an amazing picture. Microtubules. Microtubules are the largest one. All right. They form centriole in animal cells. They don't form centriole in the plant cells. They form it only in animal cells. And they are growing out from another structures that we call centrosome that exists near of the nucleus. So the centrosome is a microtubule organizing center. But not only that, he's a microtubule organizing center, but he made also a pair of centriole in animal cells. And I will show you in our big pictures. We're going to go back to my animal cells, and you are going to see them. They are here, those yellow little baby corn. Those are the pair of centriole inside of a structure that we call centrosome, which is actually an organizing system of microtubules. If you can see, they have a lot of microtubules coming out of it. So those they made those a pair of centriole. Those a pair of centriole, if I look at them very closely, if you look at them very closely, they are an arrangement actually of three microtubules each time. One microtubules, two, three. One, two, three. One, two, three. And there are 9 of them, and they go by 3. So they are an arrangement of 9 plus multiplied by 3 arrangement of microtubules. And the total, how many microtubules I have per one centriole? 27. I have 27 over here, and I have 27 over here. Remember, the animal have a pair of centriole, 2 centrioles. So all of them have 9 triplets of microtubule arranging in a ring. They can also play a role during a movement, such as the cilia and flagella. So this is paramecium ciliated. No, sorry, that's the goblet cells of trachea. And here it's actually it's not 9 triplet. This is not triplet, it's double. 1, 2, and they have a central one. So this is the difference between the centriole. The centriole, it's 9 triplet, and the flagella and cells are not 9 triplet. They are actually 9 doublet. 1, 2, 3, 4, 5, 6, 7, 8, 9. True, and they have one central pair. It's 9 multiplied by 2 microtubules arrangement over here, plus one central one. Microfilaments are smaller. They are the smallest cytoskeletons, and they help also maintain the shape of the cell, but it's also responsible of the pseudo movement where the organism stay attached actually to the surface and it start moving. And also in the plant cells, it facilitates cytoplasmic streaming. And it's you are going to see that in the lab. You are going to see the cytoplasm moving around to distribute nutriment, to distribute water to the area that they cannot be reached by those nutrients. So this is cytoplasmic streaming. It's the movement of the cytoplasm. The intermediate filaments have a diameter or size that is between the large one, this is the microfilament, or the smallest one, the macrofilament. We have the intermediate filament. Their job also maintain the shape, but it also help attaching the organs And he also, you know, what is very funny about each organs, he has this place in the cells, and around it he make what we call his space, his habitat, his location, and this is what we call lamina propria. So he maintained the lamina propria for the nucleus, which is the area that is around the nucleus. So here this table summarizes what I just explained. And then we have another structures is extracellular compound. And this is very important on maintaining, since it's extracellular, it's outside the cell, is maintain the cell-cell connections and also coordinate some cells activity. So we are going to talk about three of them. The cell wall, it exists only in the plant and some bacteria. The extracellular matrix is only for animals, and the intercellular junctions. The cell wall. The cell wall is made by a cellulose. We can find it also in bacteria, and it's made of various polysaccharides. They can be either, and because of these various polysaccharides, we can have some bacteria that they are gram positive and some bacteria that they are gram negative, depend on their proteoglycan. And we can find it also on the fungi cells. We call it chitin. And also some, most of the algae, they are made of some polysaccharide also. So it maintains the shape, he protects the cell, and not only that, he protects also the cells from the uptake of the water, excessive uptake of the water. When we study the plasma membrane and the osmosis, you will understand that the plant can take a lot of water inside without being exposed or blowing up like the animal cells do. And those cell wall protect actually the cells, and this is what it's a structural protections, and they give to the cell its rigidity. For the animal, that's for the plant. For the animal cells, we have what we call extracellular matrix. It's made of glycoproteins such as collagen, proteoglycan, and fibronectin. Collagen, those big ones, proteoglycan, and fibronectin. And you can actually band to proteins that they are embedded in the plasma membrane. They don't came inside of the membrane. They can bring whatever stimulus they receive. Like for example, fibronectins have what we call integrin receptor that is actually embedded in the membrane. It can bring information signaling a pathway and bring it inside of the to be processed by the cell during the process of signaling pathway. So they provide support, they provide regulations, they provide adhesions and movement. Cell junctions, we have for the plant, the cell junctions that exist for the plant is a plasmodesmata. Plasmodesmata are channels that perforate the cell wall. Through the plasmodesmata, some water came in, solid, and sometimes proteins and even some RNA pass from cells to cells. So they are those perforations that exist on the cell, not on the plasma membrane of the plant cell, but on the cell wall of the plant. Plasmodesmata, plasmodesmata. Okay. We have also in animal cells, we don't have plasmodesmata because animal cell do not have a cell wall. We have a tight junction, which is, this is like a membrane pressing together, are together to prevent any leaking of extracellular fluid that exists between those two cells. That are called tight junctions. Desmosis or anchoring junctions, this is fastened cells from one membrane to another. From one membrane, this membrane of the cells, fastened with this membrane of the cells. And they fasten into a strong sheet, okay? And gap junctions, this is actually more deeper. It's actually communicating cells through their cytoplasm. It's cytoplasm to cytoplasm. So this is cytoplasm that provide actually cytoplasmic channels between adjacent cells. This is membrane to membrane that they are fastened and they form a strong sheet, okay, between adjacent cells. And this is actually, they just fasten together to prevent any extracellular leaking. It doesn't, so the liquid that exists between these two adjacent cells will not go away. It will stay where it is between these two adjacent cells. Again, this is a summary for all what we talk about. And they really, really want you to get familiar with the anatomy of, this is an animal cell. And this is a plant cell. I recognize the central vacuoles. We do have vacuoles in animal cells, but we don't have this huge central vacuole in animal cells, okay? That's chloroplast. So this is a plant cells. The structure is completely, look at that. This is like a square. That's a Golgi. That's a plastid that store pigment. And that's a part of cytoskeletons. Okay. Get familiar with the anatomy. Thank you.